Hello everyone and welcome. In this video I'm going to talk about what we're doing in lab this week. And this is a lab that's actually a review lab. So there were no new readings for this week. Instead we're reviewing the concepts from chapters 1 through 4. Okay. And we're going to do some studies today looking at the topic of depth perception. So we're going to use depth perception to talk about these concepts that we have covered through chapters 1 through 4. So there's nothing new here, things that we've already done. And so what we're going to talk about today is depth perception and how we actually perceive depth. Now, basically, there are two different types of cues we can use to see depth in 3 dimensions or in 2 dimensions. Monocular cues are things that you can see with just one eye. Binocular cues are things that you need both eyes to see. So binocular cues are relying on the idea that we have 2 different eyes on 2 different places in our head, and because of that, we get 2 different images of any particular object. And how different those images are gives us information about how far away from us something is. So if we look at this picture here, this is obviously a 2-dimensional picture, and you can see some cues about depth here in the picture. One thing we have is if you look at the road in the middle, right, you can see that the 2 lines sort of converge, right, so it sort of forms a triangle into the distance, right, and as the lines converge, it tells us that that point is getting further and further away. You can also see that we have things that are overlapping each other. We know that the trees are in front of the mountains because they're overlapping it. You can see that things in the background are more blurry. You can also look at shading and light. And all of these are monocular cues, so that if I look at the picture with one eye, I can see it just as well with 2 eyes, and I can perceive depth in that particular way. Okay. Now, retinal disparity is different in that you see different images because you have 2 different eyes. And so one way to look at this yourself is hold up your thumb and put your thumb over some object sort of, 5-10 feet from you in the distance. Right? And then look at that object with one eye and then the other eye. And what you should see is that you get pretty different images based on which eye you're looking at. Right? So the disparity between those images tells our brain sort of how far away something is. So that's retinal disparity. So obviously I need 2 eyes to see those sorts of cues. Now what we're going to do today is measure your depth perception both with one eye and with 2 eyes. And we're going to do it using a depth perception box. So the box is just a simple wooden box with 2 sticks or dowel rods on a pulley. And you can see what you're going to do is you hold a string, and then the string moves those 2 dowel rods. And what you're trying to do is line them up so that they're right next to each other. Okay? So that's going to be our dependent variable, and that's going to be how far apart those 2 bars are away from each other. You can't see it on this video, but at the top of the box there's sort of a ruler that shows you how many millimeters apart those 2 bars are from each other. Now our independent variable is going to be that you're going to do this with either one eye or with 2 eyes. So we're going to do this as a within subject design, and you're going to do both. You're going to do 3 trials of this with one eye, and then 3 trials of it with 2 eyes. Okay? Everybody in class is going to do it with one eye and with 2 eyes, but what's important, because this is a within subjects design, is that we need to counterbalance. And that means that half of you are going to need to do this with one eye first, and half of you will do it with 2 eyes first. So we'll flip a coin, roll a die, play rock, paper, scissors, do something to randomly decide who's going first with one eye or 2 eyes. Okay. When doing this, when you're doing it with one eye, please use your dominant eye. So the way you can tell what eye is your dominant eye, take your thumb, put it up over an object in the distance so it's sort of blocking that object, and look at it with your right eye and your left eye. Whichever eye gives you the image that's most like your original image with two eyes is your dominant eye. All right, so we're going to do this in class and collect data and fill out this class activity form. So our class activity form here asks a number of questions. First is, what is our independent variable? Our independent variable in this case is one eye versus two eye, so using monocular cues or binocular cues to do the task. Our dependent variable is depth perception, and we operationally defined it as it's going to be the total distance between the two bars in millimeters over 3 trials. So we're going to add up the distances between those bars on all 3 trials. All right, so in any study, there's always the possibility of measurement error. And measurement error in this case could come from a variety of places. The most obvious would be you're going to have to look at the top of the box and read a ruler to determine how far apart 2 bars are, which I know you can't see the top of the box right now, but it's not very simple to tell exactly where the bars are on this measurement tool. So there's going to be a little bit of a judgment call and a little bit of measurement error in that. So in terms of whether or not you think our measure of depth perception is reliable, so a reliable measure is something that's repeatable. Right? And that is like if your depth perception is very good today, it should be very good tomorrow. Likewise, if it's very bad today, it shouldn't suddenly be very good tomorrow. So the question is whether or not this is a reliable measure of depth perception. And how could you test it? Well, what we could do to test it is look at your scores over a number of trials and see if on average they tend to average out to be about the same, if there's not a ton of variability from one measure to another. Right? That would be test-retest reliability that we'd want to establish here. Now in terms of individual differences, there are a number of sources of individual differences here. It could be like we'll have individual differences in people's eyesight, right? Some people have very good vision, some people have very poor vision. We're going to have individual differences in people's height and exactly what angle they're looking in the box at, right? How tired people are, right? Any number of things could impact people's performance on the task today. Now what's important though is we did it within subjects design, and because of that, all of those individual differences that you bring to the one eye condition, you also bring to the two eye condition. So we don't have individual differences between our treatment conditions, it's just within, and that's totally fine. Right? It just makes our data a little bit messier, but it doesn't ruin the study. All right, next is how was counterbalancing used in this experiment? Well, we counterbalance by having half of you do it with one eye first and half of you do it with two eyes first. And the reason this is important is because we might have order effects. We might have practice effects. We could have fatigue from just looking at things with one eye. And so we want to make sure that we balance those things across the different conditions. Right? Now, it doesn't eliminate order effects. All it's doing is balancing them out across the study. We didn't have to worry about random assignment here because there weren't 2 treatment conditions with different people. You were all in the same 2 treatment conditions. And so there's nothing to randomly assign you since everybody was in those conditions. So for between subjects designs, you do use random assignment to conditions. For within subjects, you don't use that. You don't use random assignment. Instead, everybody does both treatments, and then you instead use counterbalancing. Okay, our null hypothesis would be that our 2 conditions did not have different levels of depth perception, did not result in different levels of depth perception. Okay, so if we look at our data here, let me pull over the data. We have our average performance. Looking at it with one eye was 68 millimeters apart. Binocular with 2 eyes was 21.19 millimeters apart. Our standard deviations are big here, 45.73 and 31.73 respectively there. And there's a lot of variability. There's just a lot of individual difference variability in people's overall depth perception. Our t-statistic we compute is negative 10.145 with a p that's less than 0.001. This is a big difference between these two conditions. It's a difference of 46 millimeters. We can look at our effect size and see that that's an effect size of 0.99, which is a very large effect size. So we have a large difference between the groups, and this is strong, compelling evidence against the null. It's a very low p-value. So in this case, I can say that, yeah, there's a difference between the two groups, and that depth perception was better when we looked at it with two eyes, right, than with one eye. So remember, lower numbers are better here. You want less of a difference between the two bars, and so lower is better. So we did better with two eyes than with one eye. Now, in terms of whether or not we can infer causality, well, we have covariation because we have a low p-value. We have temporal precedence because our independent variable, the one eye versus two eyes, came before the dependent variable, measurement of depth perception. And we also can eliminate alternative explanations because we did a within subjects design and counterbalanced our conditions. So we don't have concerns about order effects. And the only difference between these two conditions, everything was the same. It's even the same people in our two conditions. Everything's the same except whether you used one eye or two eyes. Okay. So in this case, all three criteria are met, and so we can infer causality. Okay. All right, next question is, which of the four research goals are met by this study? So the four goals we have are describe, explain, predict, and control. Okay. So in general, with experimental studies, we're trying to explain and control, right? In this case, it's really more, I guess it's both technically, where we're trying to explain that, in fact, our depth perception is better with one eye than two eye, or with two eyes than one eye, and we demonstrate that empirically. And we can also change people's depth perception by changing how many eyes they're doing it with. That's sort of a level of control. Okay, so today we chose to do this as a within subjects design instead of a between subjects. And this was actually a very good decision here because we have a lot of individual differences in people's depth perception abilities. And so this means that all those individual differences are present in both conditions. It also, this is a very quick experiment to do. You're unlikely to get very fatigued during it, right? And I think that order effects can be reasonably dealt with when you're doing this with counterbalancing. So this seems like a good choice. It's a more powerful design because we have fewer individual differences messing up our data, right? But we also, in this particular case, have the advantage of within subject science, you need fewer people. So if we have 30 people in class, we have 30 people in both conditions. If it were between subjects design, we'd only have 15. Right? So it's very efficient and more powerful to do it as a between subject, or as a within subjects design, sorry. Okay, so we also want to look at correlations and correlating just as a review. And so what we're going to do is correlate scores on this depth perception task, and we're going to look at binocular perception with that sports experience questionnaire. Remember when we did the ball tossing lab, we had the sports experience questionnaire? Everybody's going to enter those scores into the spreadsheet, and we're going to correlate them. Right? So what I have here is our correlation matrix looking at people's performance on the binocular depth perception task with that sports questionnaire. So as a reminder, the sports questionnaire is the one where higher numbers indicate more previous sports on ball tasks, like sports with balls, baseball, tennis, volleyball, and so forth. And also, higher numbers indicate more experience. For binocular depth perception, lower numbers are better. So what we have here is an R that's negative. That indicates that as people have a higher score on that questionnaire about sports, they have a lower score on the binocular perception task. In this case, our p-value, though, is 0.46, which means that result is very consistent with the null hypothesis, that there's really no correlation there between these. That 0.10 or negative 0.10 is probably just due to chance factors. All right, so this was a very small correlation, negative 0.10, and negative means that as one goes up, the other one goes down. In this case, though, remember that the negative correlation means that as performance on the questionnaire got higher, or that people had more experience with ball sports, binocular depth perception, the score got lower, which is actually better, right? A lower score is better here. So our evidence is very consistent with the null, and all we're looking at here in terms of the 4 goals would be description or prediction. You could describe the correlation between things and determine if one is a good predictor of the other. Now in this case, can we infer that sports experience causes better depth perception? Well, no, and we can't for several reasons. One is that we don't even have covariance in this data set. There's no relationship between these 2 variables. We also don't have temporal precedence. We don't know if the better depth perception came before the sports or not, the sports experience. It's entirely possible that people with better depth perception are more likely to play ball sports. Or another way of thinking about this is like people with a really, really terrible depth perception are unlikely to want to play sports where balls are getting hurled at their head if they're not very good at perceiving that depth. Right? So we don't have temporal precedence, we don't have covariation, and so we cannot infer causality here. Right? It's very possible that other things are causing this relationship, even if it existed. Right? We don't. Our data is very, very weak, suggesting there's not even a correlation there. Though I don't know what's going to happen in class. This is just data I had to make up for this particular video here. Okay. All right, so if we go back to your depth perception slides here, in the terms of the group's practice quiz this week, it's a little bit different in that you're going to have a practice quiz that's a little bit longer, more like a test length practice quiz, and there's no final quiz. So the group quiz is going to be worth 20 points rather than the usual 10. As usual, you get to take it as many times as you like, and we're going to record your highest score. But really focus on, think of this as a practice test, and you need to understand these concepts. So don't just try to get the right answer. Work with your group, work with your instructor to make sure that you understand why the answers are what they are. Okay, so that's all I have for this video. And if you have any questions about the exam, be sure to let us know.